Good morning. Welcome to the 597th Biweekly Assembly of New Asia College of CUHK 2016 to 2017. We will now sing the college anthem to commence the assembly. Please stand up. Please be seated. Students, please be reminded that you must punctually attend the biweekly assemblies and colloquia which you have registered for. Students admitted in 2012 to 2013 must register for and attend 2 biweekly assemblies, while students admitted in other academic years must register for and attend 3 biweekly assemblies or 2 biweekly assemblies plus 1 student lunch or dinner colloquium. Should students fail to register for assemblies or have any inquiries to the attendance, please contact the Dean of Students' office. Before entering the hall, please make sure that your attendance is successfully recorded by the card reader. A successful record is signified with a beep sound and green notification light. Students can check your attendance record on the Registration and Attendance System, or RAS, after 4 p.m. on the next working day after the biweekly assembly. The 597th Assembly of New Asia College of CUHK 2016 to 2017 will start now. Today's assembly is the first lecture of the 3rd Yen Kwok Yung Lecture in Life Sciences, jointly organized by New Asia College and School of Life Sciences. We are pleased to have Professor Peter Holland from the University of Oxford sharing with us on the power of evolution and the path to scientific discovery, 進化的力量及科學發明之路. We are also pleased to have Professor Chu Ka-ho, our Associate College Head, to introduce the speaker and to be our moderator. Now, may we invite Professor Holland and Professor Chu to come up on the stage. Professor Holland, please. Professor Chu, please.
Professor Holland, fellow alumni, colleagues, students, ladies and gentlemen, good morning. So in this assembly, we'll have the first lecture of the Yen Kwok Yung Lecture in Life Sciences. This lecture series was initiated 3 years ago, and we are honored to have very distinguished speakers over the years. Professor Yan, to whom this lecture was in memory of, is the founding chairman of the Department of Biology of the New Asia College. He is actually a very famous Chinese zoologist working on birds. So this year, we are honored to have invited another very famous zoologist, Professor Peter Holland to deliver this year's lecture series. Professor Holland graduated in 1984 at the University of Oxford as the top student of zoology. 3 years later, he received his PhD in genetics from the University of London, and in 1994, he was appointed Professor of Zoology at the University of Reading, becoming the youngest full professor at the United Kingdom. In 2002, he returned to Oxford to become the 11th holder of the prestigious Lennery Chair of Zoology, founded back in 1860. He was elected to the Fellow of Royal Society in 2003 and served as the head of the Department of Zoology at the University of Oxford from 2011 to 2016. Professor Holland's research focused on the evolution of animal genomes with a special interest in the origin of new genes. The genes that he discovered from the genome of human and animals have high relevance to stem cell research and other biomedical research. Professor Holland has been awarded numerous prizes and medals for his research. Just to name a few, that includes the Linnean Medal, Genetics Society Medal, and the Scientific Medal of the Zoological Society of London. So today we are grateful to have Professor Holland to give us the talk on the power of evolution and the path to scientific discovery. And last but not the least, I also like to take this opportunity to pay tribute to our donor, Dr. Yam Kai-ming, who is the daughter of Professor Yan, and her family for the endowment of the Yang Guoyong Memorial Fund, which makes this lecture series possible. Thank you. So now may I give the floor to Professor Holland.
Okay, good morning, everybody. And thank you, everybody, for coming. And certainly, I'd like to thank UH College and the School of Life Sciences for inviting me today. I want to talk about a few different things today. And this talk is going to be split into 3 different parts. First of all, I want to talk about the subject of evolution. What is evolution? Why should we be interested in it? Whether we're scientists or not, why is evolution such an important topic today? And what I want to try and show you is that evolution really is the glue which holds the whole of the life sciences together. And without our understanding of evolution, we really wouldn't understand life sciences. But also I want to show you that evolution has our knowledge of evolution has applications to real-world issues. Secondly, I want to sort of introduce you to some of my own research, and hopefully a little bit of an insight into what I've been doing for the past 20 or 25 years. There may be some interest there about some of the decisions that I've made in my career, which might be of interest to other people. And then finally, I'll end with a few words of advice, which you can either listen to or ignore as you wish. So let's start with evolution. Now, of course, evolution is the real explanation for that whole diversity of life. We can walk around outside and we see animals and plants and fungi, and if we have a microscope, we'll see bacteria, and we can look in great detail. And that vast diversity of life on the planet is the product of evolution. But what do we really know about evolution? I think there's a little bit of a problem in, I think, the general public's understanding of evolution and perhaps some aspects of evolutionary education, which is unfortunately, whenever we talk about evolution, many people think, well, this is an old-fashioned subject. We think of people like Charles Darwin and Alfred Russel Wallace and work they did 150 years ago. and they wrote famous books, which, of course, people still talk about. But really, I want to convince you that evolutionary biology has changed. Our depth of understanding of this subject is really something quite dramatic today. And let me try and explain why that is. So let's think about how evolution works, the framework which was put together 150 years ago. So here we have, I want you to imagine that each of these squares here, each of these small squares is, let's say, an individual, an animal in a population. And a variant arises. So one square there is a different colour, that's a variant. Okay, something has changed, that individual is different. Then what happens is, because that individual may be better adapted to its environment, it may leave more offspring, and gradually the numbers of that variant increase in the population. And through that process called natural selection, changes are driven through populations, which over a long period of time, many millions of years, gives rise to the diversity of life that we see today. Now that sounds very straightforward, but just let's think about some of the details involved in that. Let's look at the first one, variation. What is variation? What do we mean by that? What we really mean is mutation in DNA. Variants are mutations in genes. So, of course, understanding DNA sequences, understanding genes, is actually a modern scientific research area. It doesn't bear a great deal of relation to what Darwin and Wallace were talking about because they didn't know about DNA, they didn't know about genes. So today, if we really want to understand this part of the story on the left-hand side, this is a modern research, scientific research area. Well, let's look at natural selection. What is natural selection? Natural selection, really, is the relationship between organisms and their environment. Why do some of these variants fit better to their environment than other variants? That, of course, really is ecology. And ecology also is a modern scientific research area undergoing a great deal, in which there's a great deal of interest today. And what do we mean by adult form? How do these variants, how do these genetic changes lead to changes in adult form? Well, really, this is a question in developmental biology. It's a question in how cells talk to each other. It's how cells get organized in the body, and how the embryo forms from a single cell all the way through cell division and cell communication to give the final body form that we see in an animal. So that is developmental biology, and again, that's a modern scientific research area. So I think what I've tried to show you there is that the study of evolution, when we think about evolution, we think about evolutionary biology, we shouldn't really be thinking only about those important contributions of scientists long ago. We should be recognizing that this is a modern scientific subject with a great deal of excitement in it. But what I want to look at now is that maybe that's a sort of explanation of why evolutionary biology is interesting, but that's a little bit different from asking the question, why is evolutionary biology important? So what I want to do now is just spend a few minutes thinking about why the study of evolution is important to our lives today and the modern world today. And in doing this, I'm going to pick some examples from across science, not my own research, but research of a number of other scientists to give you a little flavour of this. So the first reason is that we can learn from nature. All that product of diversity out there is the product, all that biodiversity is the product of evolution. If you walk outside this lecture theatre and you see a butterfly flying, or a bird flying, or a bee, or a fly, just pause for a minute and watch it flying. And even in a few seconds, you'll recognise that those birds and insects are so much better at flying than any of our inventions that humans have made. Better than any aeroplane, better than any helicopter, better than any drone. And why is that? It's because through millions of years, a whole variety of different modifications have happened to fine-tune how those animals actually fly. What I'm showing you here is some research from two colleagues of mine Graham Taylor and Richard Bomfrey at the University of Oxford, who've been studying how birds fly and how insects fly using a whole variety of different techniques. The video that was scrolling in the middle here was an internal view of a fly flying. So I'm actually going to run that again. What you're seeing there is the internal workings of the muscles contracting inside the fly as it's actually flying, to understand the forces involved in producing those very finely controlled flight movements. The other diagrams here are showing evolutionary variation in wing pattern, but also aerodynamics of different bird wings. Those sorts of studies, and I won't go into any detail, but those sorts of studies are already proving very, very useful to people in the aeronautics industry who are making modifications to design of aeroplanes and design of drones to improve the efficiency of our own man-made vehicles. Let me give you another example where we can learn from nature. This is the work of a colleague of mine, Pat Nuttall, at the University of Oxford. Now, what Pat works on are these small animals here. This is a tick. Now, ticks are not very pleasant animals. They are ectoparasites. They bite us. They transmit diseases. But they're also the product of evolution. And there are some remarkable adaptations that ticks have. One of which is if you are bitten by one, there isn't actually a great deal of inflammation in the tissue. And that's because through evolution, ticks have evolved proteins to suppress inflammation in their host. We can study those molecules, and some of those molecules are already being copied for use in medicines for treating inflammation problems, such as arthritis. So again, we can learn from evolution. Another reason why evolution is important to our modern lives is because we can learn from our enemies. What I mean by our enemies are things like bacteria and viruses, which cause so many problems to our health. Those organisms, or those viruses, are also evolving, and they're evolving very, very quickly. And if we can understand how they evolve and understand how their DNA sequences evolve and their RNA sequences, we can understand how to combat them. So what I'm showing here is work of my colleague Oliver Pybus at the University of Oxford, who's been using that methodology to not only work out where outbreaks of disease have come from, but also to predict where outbreaks of disease will come from. Now, those insights would not be possible without us understanding how DNA evolves. A third reason why evolution is important to our lives today is that we are the product of evolution. Now, we can see this, of course, if we just look at our anatomy. It looks so similar to the anatomy of many other animals. But let's look right inside. Let's look at how our genes control the formation of the body. Let me give you a very famous example. There's a set of genes, very well-known genes, called Hox genes. And what Hox genes do is tell cells where they are along the body, from the head to the tail. They sort of give a little address to cells along the body. And that's very important because the cell needs to know where it is so it forms the right structure as an embryo grows. Now, these genes were first discovered in flies, and you can see here, here's a fly, and this complex diagram down at the bottom just shows that these different Hox genes are expressed at different places to tell cells where they are in a fly, or at least in a fly embryo. But they're also there in many other animals, including in humans, and they're doing essentially the same job in our embryos, telling cells where they are along the body. That's because we are all related by evolution. Humans, like almost all other animals, use those sorts of genes to tell cells where they are. So again, this is because of evolution. Then the final reason that I want to emphasize is perhaps the most important of all of them, which is we must never forget the importance of pure research. Sometimes research scientists are not out to solve a particular problem, they're out to investigate the workings of the world, the workings of either life or the universe or some other issue. And we don't know where that research is leading to. It's like walking across this bridge. It looks very interesting, but we don't know what's at the other side. That's really what pure research is all about. And I'm going to just give you one example of that, which I think is such an exciting example. And it's work from a whole variety of scientists around the world. Two of the most important people involved in this research were Philip Ingham of Oxford and Singapore and Exeter, and Matthew Scott in the USA. And what they were working on sounds very like a research project where you don't know where it's going. They were interested in how a fly makes the stripes in the fly. Now, what I mean by the stripes are really the segments. If you look at an insect, any insect, you will see its body is divided into different units. But how do those units form? They form because cells are talking to each other in the embryo. This is the embryo of a fly here, and different cells have to talk to each other. They have to send molecules to each other to work out where to make boundaries or stripes in the embryo. And the work of these scientists and many others deciphered the molecules which are involved in that process. Genes such as this gene here called hedgehog, which signals to this receptacle patched, which inhibits this receptacle smoothened. Great deal of biochemical knowledge. Now, why would that be of interest to us? Why would that be of importance to our lives? Well, it turns out that that very same signaling mechanism, those very same molecules, are used in us for all sorts of different processes in the body, all sorts of situations where cells need to communicate. And it turns out that one of the commonest human cancers, a common skin cancer called basal cell carcinoma, which is the one induced by sunlight, is caused by disruptions to that very same biochemical pathway. And because of the knowledge which was gained from working on the flies, drugs could be very quickly developed as medicines to treat that type of cancer. So to go back to here, we would never have known, those scientists would not have known where that path of pure research was leading, but you can see how important the conclusions can be. And that's all because of all those insights I've given you are because of evolutionary biology. Okay, so what I've dealt with so far is I've tried to show you that evolutionary biology is a very interesting subject and a very important subject, but I haven't drawn on my own work. I've talked about the work of others. What I want to do now is spend a few minutes talking about my own work and the scientific projects that I've been involved in. And in particular, I want to talk to you about how my interest in this topic, because I would call myself an evolutionary biologist, how did my interest in this topic come about? And I could start by just going through a sort of formal biography of saying, well, I was born here and then I did this and then I did that. I'm not going to do that. That's all a little bit boring. I want to give you a few different sorts of insights. Let's start right at the beginning. This is me, looking a little younger. So my interest in science and my interest in life sciences really just started quite simply with an interest in animals. As a child, walking around the woodlands, walking around the fields, catching insects, watching birds and mammals, and studying what they did. It's just a fascination of mine, nothing more than that. But a fascination that really occupied me wherever I went through my childhood, through woods and fields, rivers, collecting insects, etc. So that was my interest. I went to school at this school, high school, at a school near Manchester in the north of England, which was a very normal, ordinary school. This is a picture of it. You can see it's not particularly architecturally very exciting. And of course, I studied a whole variety of different subjects there. The subject that interested me most, of course, was biology. Although I have to admit, and maybe some of you sort of might understand this, the teaching of biology in high schools was not quite as exciting as I hoped it would be. It didn't really capture my imagination in quite the way that really the natural world does. We walk out there outside these doors and the natural world is so exciting, but unfortunately some of the textbooks of biology are a little bit dull. But anyway, I studied those subjects and decided, of course, that I would go to university and study biology. But where to go? Well, one of the most famous universities in the UK is Oxford University, and it has a very good biology course, and I decided that I would try and apply to go to Oxford. It was not a very common thing to do from my school. I don't think anybody in the history of my high school, had ever been to Oxford. So I spoke to the science teachers at my school and said, well, I'm thinking of applying to Oxford. You know, it's a very good university, and they've got a very good biology course, so I'd like to go there. And the head of science, he took me quietly to one side of the corridor. Actually, I can tell you, it was exactly this corridor just up there. He took me to one side and he said, you're wasting your time, you're wasting the school's time, and you're wasting Oxford's time. Now, that's not the most encouraging thing to say to a student, but I just ignored it. I did apply to Oxford, I did get accepted by Oxford, and I went to Oxford as a student, and I've never been back and spoken to the science teacher again. But I had a good time at Oxford. We see pictures of Oxford like this. I'm sure we've all seen pictures like this, they're on postcards and books. That's not the reason I wanted to go there. I wanted to go for this. This was the zoology department. A brand new building, lots of exciting biology was being done there in research and in teaching. So I wanted to go there. I had a very exciting time as an undergraduate, as I'm sure you're all having an exciting time as an undergraduate, because learning is such an exciting thing to do. And in studying biology, I began to realize that there were actually two areas of biology that really excited me. The first one was molecular biology. The structure of DNA, genes, proteins. I thought this was so exciting, the genetic code. The other area of biology which excited me was the life around us. Just like I'd been growing up collecting insects and catching fish and watching birds, the diversity of all those animals was so exciting. But to me, they seemed such different parts of biology. Genes or animals. What do I do with the rest of my life? Because I decided that I would try and do research and study do research for a PhD. But I felt I had a really big decision to make in my life. And I think we all face decisions in our life. And sometimes we agonize for a long time about those decisions. I'm going to show you in a minute that we really shouldn't worry so much about these decisions. But I did. I worried a lot about, should I do a PhD in molecular biology, or should I do a PhD on animals? Well, I decided, I came up with a very practical solution. I thought, well, if I do a PhD in molecular biology, I can always keep on studying animals as a hobby. If I do a PhD on animals, I can't really do molecular biology as a hobby because I can't have microscopes and gels and things in my house, so that's not going to work. So I decided, okay, I'll do a PhD in molecular biology, but first of all, I need to get this sort of interest in animals needs to get out of my system a little bit. So I decided to go to South America for a few months. And this is me, age about 20, in the rainforest, collecting insects. So I went to South America, to Venezuela, just about here, to explore rainforest and to collect insects. And in particular, I wanted to collect some very unusual bees, which were called euglossine bees. I won't tell you about their biology now because we'll be here all day, because they're fascinating insects. But it was a very exciting time. And I came back after being to South America and thought, well, that's fine. I've now done my animal part. I can now really concentrate on molecular biology. And I went to do my PhD in this research institute here in London, the National Institute for Medical Research, a grand building in the north of London, which actually has just closed and relocated to central London. My PhD supervisor, Bridget Hogan, here, there's me, ran a research group working on the genes expressed very early in embryos. And I wanted to study those. I really wanted to isolate genes. I wanted to sequence DNA. I wanted the excitement of being the first person in the world to see A, C, G, T, G, C of a particular gene, which of course it was possible to do. And as a student, the first thing I did was to work on those genes I mentioned a few minutes ago, those Hox genes that tell cells where they are along the body. And what I was able to do, along with a lot of other people, was show that what these genes were doing, they were present in lots of animals, and they were doing a very similar job in different animals. And then something dawned on me. If we can apply molecular biology to these sorts of animals. We can apply molecular biology to any animal. There's no reason why there needs to be a decision here. There's no reason why we need to think molecular DNA or animals. It's all the same thing. The technology was available to take the methods of molecular biology, which had all been developed for bacteria and cells and model organisms, and actually apply them to any animal at all on the planet. So with that excitement, I thought, well, this, as a biologist now, we can do anything. We can think of any animal that excites you and start studying its genes. So at the end of my PhD, I was fortunate to be offered a junior teaching post at the University of Oxford, back in the department that I came from as an undergraduate. And I had to make a decision as to what my own research project would be. So I decided I was going to work on the genes, the molecular biology of this animal here. Now, many of you won't recognize what this animal is. It's a little animal called Amphioxus. It's about 2 or 3 centimeters long, and it's marine. It lives in the sea. So why would I want to dedicate some years of my life to study the genes of this small animal? Well, it was suggested to me that maybe the reason I chose this was because they lived in places like this. They're very common off the coast of Florida, on the Gulf Coast here, on beaches like this. And that actually is where I went to collect the animals and study them. But that's not the reason I chose them. The reason I chose them was because of evolution. These animals, the Amphioxus, sits at a very important place in the evolutionary tree. It sits really just at the junction, just at the difference between the invertebrates and the vertebrates. The vertebrates are the animal with backbones, like mammals, birds, fish, etc. And the invertebrates are things like worms and flies and snails. And this animal is right at the boundary between the two. So if we could understand how genes work in this animal, I thought maybe we can get an evolutionary insight into this really important change in evolution. So that's what I opted to do. And I went out to Florida, which really didn't look like that at all. It looked more like this. There were hurricanes, there were sharks, there were stingrays. But we could dig in the sand, we could collect these animals, we could collect them, we could obtain their embryos, we could extract their DNA and start sequencing their genes. It was a very, very exciting time. And lots of discoveries came from that. I'm not going to go through those discoveries in detail. I just want to give you a little flavour of the sorts of things we were doing. So first of all, we looked at those Hox genes again, the ones that tell cells where they are. And we discovered they were quite a bit different in this animal from in the vertebrates, like humans and birds and fish, etc. Because this comparison revealed something quite interesting. A rather large change had happened in genes in evolution on the way to the vertebrates. We were able to work out how these genes were regulated in Amphioxus and get a view of what they were doing there. But also we found some unexpected things. We didn't just find the Hox genes. We started discovering other sorts of genes, which although present in other animals, including humans, hadn't really been well studied before. So we discovered a group of genes which we call the para-Hox genes, which do something slightly different in the development of the gut. And they're present in humans as well, but they really hadn't been recognized in the same way. So we learned a lot about evolution from those studies, but there are a couple of other things that I want to sort of take home from what I've just said. First of all, was that decision between should I become a molecular biologist or should I become a zoologist? What I've tried to show you is that actually there was no difference there really, because the technology of one subject could be applied to another subject. And I think the general message from that is that sometimes those big decisions in life are not one or the other. And maybe we worry too much about some of those decisions. We should just do what we think is right and what we think is exciting and what we think is interesting. And I think things should work out if we stick with that philosophy. The other thing which is quite quite important, but it's rather hard to sort of articulate, which is that when I said to my friends and colleagues that I'm going to work on this animal here, Amphioxus, I have to say my decision was met with a little bit of puzzlement, a little bit of bewilderment, Because at that stage, molecular biology was really only being applied to the main animals which were studied in laboratories, like mice and rats and flies, not strange marine worms. So even though that work turned out to be very successful, it was only successful really because we were doing something different. And I think it's really important to say, don't be afraid to do something different in the decisions in your life. Follow your own ideas. And if people look at you with a bit of puzzlement, look a bit bewildered, that's their problem, that's not your problem. Be ready to follow different ideas. So around that time, after a few years at Oxford, I moved universities. I moved to the University of Reading, also in the south of England, and then back to Oxford again a little while later. But something interesting was happening in biology at the time. There was a real revolution happening. We've all heard of the Human Genome Project. The Human Genome was first published, the first draft of the Human Genome Sequence, all the A's, C's, G's and T's of the human genome in 2001. Now, there was a lot of excitement at the time about what that meant for human biology, but I was excited in it for what it meant for evolutionary biology. Because maybe now the technology was moving so fast, the technology to sequence DNA, that maybe now we should not just be looking at one gene at a time, but maybe we should be looking at all the genes in the genome, because the power of sequencing DNA had moved on so fast. So to give a little idea of a scale here, if we know that genes, DNA is made of A, C, G, T, etc., again and again, one gene on average would be about 3,000 of those long, but the whole genome, all the genes in an animal, whether it be a human or something else, could be about 3 billion of those. So we're talking now about a problem 1 million times more complicated. Okay, we're looking at all the sequence of the DNA, but technology was moving fast. So I really felt it was time to embrace that technology and start trying to use the new technologies of whole genome sequencing to study evolution. Of course, many people actually around the world were doing exactly the same thing, because I think what you have to be able to do in science is keep an eye out as to what else is happening around you, particularly technology that's changing. New technologies can come along. We need to be ready to grab those technologies and use them for our own questions. So the first application of the whole genome biology that I did was to say, well, which genome has been sequenced? Is the human genome? Looks a very interesting genome. I can go and look in the human genome with my students and try and analyze the particular genes that I'm interested in, which is the Hox genes, and a group of genes related to them called homeobox genes. So there's a few hundred of these genes, and analyze all of them. We did that for the human genome, but that set the framework for us to study them in an evolutionary context in other animals. So first of all, we, together with many, many colleagues around the world, we were able to sequence the complete genome of Amphioxus, that small marine animal, and look at all its genes. And then through collaborations, I've been very fortunate to be involved in analysis of the DNA sequences of many, many other animals. There's just a few here: butterflies, parasites like tapeworms, and animals of interest to aquaculture like the Pacific oyster. So being involved in these consortia, helping large groups of people analyze these very complex sets of data, has been a very exciting thing to do. But in general, putting all that together, if I look back and think, well, what's been the point of all this sort of research? I think really there's a very, very clear thread there, which is if we go back to that introduction I gave you a few minutes ago about evolutionary biology, and what is evolutionary biology? What we're really talking about is how changes in genes lead to changes in how embryos develop, which lead to changes in what organisms look like. And all this analysis of genes in different species has given us much more of an insight into that pathway from genes to development to the adult form, and how that changes in evolution. Okay. What I want to move on to now, very finally, is a few general words of advice. And I'm just going to give you 3. These are meant to be words of advice to any to any young student, really. Whether you're a scientist or not a scientist, it doesn't really matter. But I'm going to give you 3 words of advice that I really want you to think about, which are these 3: chance, criticism, and confidence. And I'm going to explain a little bit about what I mean by each of them, because in my own experience, all 3 of these have been very, very important in making the right decisions. The first one is chance. We tend not to think very much about chance. But chance is really important in discovery, whether that's scientific discovery or any other form of discovery, whether you're a historian or geography or anything. Chance is quite important. That doesn't mean we should stumble around not knowing what we're looking for. It just means that in our studies and in our research, maybe in all areas of our lives, we should be alert for the unexpected. Be ready for the unexpected. Because the unexpected is the thing that nobody else has seen. That's what we mean. If we only go out there and find the things that have already been known, all we're doing is adding detail to an issue. Well, that's fine. Detail's important. But if we're always keeping an eye out for the unexpected, then we can find new things. We can discover completely new things. Which is the opposite of this. If we have heroes in our fields, like heroes in science, we can have anti-heroes, I think. My anti-hero is Henry Gantt. I'm sure many of you will have seen a Gantt chart or used a Gantt chart. A Gantt chart plots out in time what you're going to do every week or every month from the start of your project. Decide title of thesis, work out thesis, gradually do this until end of thesis or end of research project. Now, if we all did that, life would be a little bit boring. What you really want to be doing, the ideal Gantt chart, will be to go part of the way through this and then say, oh, I've just seen something really unexpected. Didn't expect that at all. I'm going to change. I'm not going to do my original project. I'm going to do something else. Okay? That's why I don't like Gantt charts. Oh, we all have to do them, I know, but for any project. But be careful. Do not be tied by them. Much more important is to keep an eye out for the unexpected and take those chance opportunities. I'll give you just one example from my own work. I mentioned this group of genes which were controlling the development of the gut, which we found in Amphioxus and are also present in other animals. We weren't looking for those at all. We found them by chance. We were looking for something else, and then we saw a result in the lab and we thought, that's funny. I didn't expect that gene to be there. So we stopped what we were doing and chased that and found something new. That's what I mean by chance. The next word, criticism, is a really hard one. It took me a long time to accept this, that this was important. It's a very, very hard lesson to learn, which is that criticism is a good thing. Criticism is not a bad thing. It's a word which, in English anyway, has a very negative meaning to it. To criticize something or to be critical sounds like we're making out that it's bad. But actually we're not. We're trying to make it better. And that's a really important thing. So what I want to encourage you to do is to embrace criticism. If somebody in your life or in your work and in your studies criticizes and says, well, this isn't quite good enough or this isn't quite right, think about it. Just stop and think, well why? What does that mean? And can that help me? Now, as I said, that was a very hard lesson for me to learn. I mean, as a scientist, we publish scientific papers, we write research funding applications, grant applications, and every time they're sent out to other scientists who tear them to shreds. They criticize them, they write comments, they send pages and pages of critical comments. And at first, when I was a young scientist, I was very upset by these. Because, I mean, how dare these people criticize my work so badly? But actually, they're making constructive comments. And it's the same in many other walks of life. It's how life works. It's how we should learn. So, not only am I encouraging you to take criticism, to really think about it in a positive way, but I want to encourage you to give criticism as well. To your friends, to your tutors, to your lecturers, to your professors. Think about it. You have to think, because criticism can be given in a positive way. It's not a negative thing, it's a positive thing. And I always want to be, if you're always thinking in that way, you can start to challenge things, and you can start to think, well, how can we change things? How can we make things a little bit better? Or maybe we can think about this problem in a slightly different way. So it's a very difficult one to quite get right, but it's a very important one, because if we're not critical, we won't move forward. The third one is confidence. And again, this is quite a difficult one, because I don't mean arrogance. I don't mean people should walk around thinking, well, I'm confident, I'm going to be right. What I mean is, sometimes, once you've taken on board advice and criticism and thought about it carefully, then you can make a decision. You can make a decision which is your decision, a decision that you can stick to. Remember this one here, where my teachers at my school told me I was wasting my time going to Oxford. What I had to do at that situation was think about it carefully and think, well, are they right? Actually, we know they were wrong. But I had to make a decision, a careful decision, about what they were saying and what it meant to me. And I'm going to give you one final example, and it's an example from my own research, quite a recent example, when confidence was really, really needed in my own research. And it comes back to that work I mentioned on genomes and analysing genome sequences. When we started to analyse the human genome sequence, to analyse all these genes called homeobox genes in the human genome, we described all the homeobox genes. This is together with this work was done with a graduate student in my lab, Anne Booth. We analysed all the homeobox genes in the human genome, and we discovered some new ones, genes that nobody had seen before. Very exciting. We gave them names. But here's a picture of Anne and the genes that we discovered. New genes. We didn't know what they did. We had to invent names for them, and we had to make a few hypotheses. We learned a few things about them. We proved that these particular genes were not only found in humans, but they were found in all the mammals. If you go outside the mammals, they're not there. But for 10 years, we did not know what these genes did. Every experiment we tried failed. We wrote scientific papers, which were not very well received. We wrote funding applications, which were terribly received, because we didn't really have any clues to what these genes did. Many of my colleagues were telling me, just don't even try to work on those genes. They do nothing. They're just junk in the genome. They don't do anything. But just a few years ago, we did find out what they did. That those genes are very important genes. They're expressed in the human embryo very soon after fertilization, just when the embryo is just a few cells, about 8 cells to 16 cells. And then the genes stop working, and they're never used again for the whole of the rest of your life. Whether you live to 100 years old, these genes don't do anything ever again. They were just used just from 8 cells to 16 cells. So they have very important roles. What we showed is, I don't want to go into the details, but these genes control the activation of other genes in the very early embryo. So we really had to be persistent with that. We had to be confident. We had to be not afraid to fail and to take a few risks. And I think that's a general lesson about our lives, whether as scientists or as students of other subjects, or indeed in our general life. Once we've decided what we want to do, we should have the confidence to follow it through. Please do not be afraid to fail, because if we fail in some of our ideas, that means we're taking risks. Other ones will work. Some will fail, some will work. That's absolutely fine. So I'm going to stop there. I hope some of that has been of interest to you. The 3 parts of my talk about why evolution is important, my own journey in the subject, and a few words of advice. And of course, I'd be very happy to answer any questions. Thank you.
Let us again express our gratitude to Professor Holland for his wonderful lecture with another great round of applause. Now, may we invite Professor Chu to hold the Q&A session. Professor Holland, please. Professor Chu, please. Please feel free to ask any questions.
Thank you, Professor Holland, for this very stimulating lecture. Well, I think that will raise a lot of questions among our audience. So, are there any questions? Okay, over there. There's a microphone at the back.
Testing. Hello, Professor Holland. My name is Chester. I'm from the Faculty of Business Administration. I'm a year 3 student. First of all, thank you very much for the really intriguing talk today. And I now have a question about human beings. So, there are a lot of technological advancements today, like there are better transportation, so we can walk less. There are computers, so we can remember less. So that being said, all those things are the evolution on technologies, right? And people are worried about if this goes on, actually, we human beings do not need such a strong body as before, such a large brain as before. So it is actually devolution happening on us human beings. So do you think these kind of things will happen? Sorry, I'm a little bit nervous. Do you think these kind of things will happen? Are you worried about this change? Thank you.
Okay, that's a very interesting question. I don't think I'm worried about this. I think at the moment, maybe we're only just seeing the start of the future, but I think the changes that you've talked about in terms of transportation or computers still seem to me to have quite small impacts on the way we use our attributes. We still have to use a lot of brain power to use a computer, or at least to use a computer very well. So personally, I'm not so worried about that. But it's a very, very interesting question. I think let's revisit it in 100 years.
Okay, there's a second question.
Hello, Professor. I'm a science student in year 1. I would like to ask us whether the ideas of revolution can use beyond the biology, such as political or moral or something other else. As you say, that is not very different in every different subject, as molecular biology and the insects are not very different. Or maybe it seems that that is no different in biology and the other subjects. Or the ideas of evolution can also be used on the other subject. Thank you.
Okay, so the question is whether ideas of evolution can be applied to essentially non-biological subjects. I think yes, but we have to be a little bit careful because evolution involves variation and inheritance of that variation. So only in some of the examples that you mentioned do we have both those two things happening, the variation and the inheritance of that variation. Some of the cultural ideas, of course, we can talk about cultural evolution. An idea may arise and then spread through a population. It's spreading in quite a different way, of course, because it's spreading from person to person horizontally, not necessarily person to offspring vertically. So the mechanism is a little bit different, and the consequences that will therefore be a little bit different. But I think, yes, we can use the analogies. We just have to be a little bit careful with the analogies because the process is different.
Yes, you have to go to the back. Mr. Wong is one of our trustees of the college.
Professor Holland. I'm not a student. I'm actually a graduate of the college. Well, thank you for the talk. Actually, I don't quite understand a lot about what you said about molecular biology. But just a question on the evolution of human beings. How do you find our students today different from the students, say, 30 years ago, 50 years ago, or even 100 years ago, in terms of intelligence and capability, from your point of view?
Well, I can't talk from personal experience of students of 50 or 100 years ago. Maybe 30, just about. But of course, there's plenty has been written about students and learning over that period of time. So we should be able to say something. I don't think there's any difference in abilities and aptitudes, or even really attitudes. What is different, and what is the challenge today, is that our fields of inquiry are so large. There's so much is known, and it's so broad, and there's so much to think about, that it's actually very difficult for students of today to have a very broad understanding. The students become more and more focused on particular areas. Now, I don't know if that's a good thing or not. I think time will tell. I really don't know. But it's an inevitability. We can't, with so much knowledge, and there's so much accumulation of knowledge over the past decades or century, that none of us can be experts in everything. That probably wasn't, 100 years ago, the scientific world was much smaller, and I think it was possible to have a pretty good appreciation of the whole subject. That really isn't possible today for any one person. We have to recognize that, and therefore students have to make some difficult choices about what to concentrate on and what not to concentrate on.
Maybe I have a follow-up question. So, as an Oxford professor, you must have a lot of students approaching you for postgraduate studies. So, how do you pick students? What kind of attributes are you looking for?
The most important attribute is an inquisitive mind. That's clear. Not really how much does this person know, more how much does this person want to know. How inquisitive are they? How curious are they about the world around them? That's usually quite clear to see that when talking to somebody. And I think that's a really important attribute.
Yeah. Okay. There's a question on up there.
Yes.
Yeah. Please.
Hello, Professor Holland. I'm a Year 3 student of molecular biotechnology. Excuse my poor English here. But anyway, I really want to ask you about something, and that's a small group of advocates in mainly the United States, I think, but actually in all parts of the world, that denied evolution as a scientific theory and instead promote intelligent design as a quote-unquote alternative. How do you think is going to happen? Is it an impending disaster for the scientific community, or is it just another minor obstacle for us in the future? Thank you.
Well, there's sort of two parts to that question, really. I come. The UK is a slightly different sort of country. I mean, we have Charles Darwin on our banknotes and on our coins. He's a national hero. So the study of evolution is something seen as central to our culture in the United Kingdom. I recognise it's different in different countries, and particularly, I think, in the United States. I think as a scientist, it's a very easy argument to make, because evolution is a testable science. We can make very, very clear hypotheses and very clear predictions and test them. We do this all the time, and this has been done thousands of times, and every time the hypothesis is proven. So, to my mind, it's no different from, let's say, a belief in gravity. I know if I hold a pen and I let go, it will go downwards, not upwards, because of all the other tests of gravity. To me, evolutionary biology is exactly the same. I can hypothesize what a gene sequence will look like in a particular organism because of what it's related to, and when we sequence the gene, that's what we find. So it's the same. But the second part of your question really was whether it's a worry, whether it's a problem, whether it will hold science back. I think that's quite a genuine problem, maybe not in a country where I come from, the UK, but in some countries that might be a problem, because it's a question for the politicians to make decisions about where funding goes and to recognize that life sciences is built on an understanding of evolutionary biology. If we don't have an understanding of evolutionary biology, we can't do life sciences. So that needs to be recognized by the funding agencies and the politicians. So from that perspective, yes, it is a worry. And we need, I think as scientists, we have a duty to make clear what our level of understanding is of all our subjects.
Yeah, and that is actually the purpose of this kind of lecture series. When we talked to the donor 3 years ago with Professor Wong, college head, well, that's the idea to invite famous scientists like Professor Holland to come to college to let the students and the general public to know more about life science and evolution. And we hope that we can achieve this goal. Okay, there's another question.
Hello, Professor Holland, and thank you very much for inspiring speech. Since one of your major research areas is about genetics, so I really wanted to know your personal attitude toward transgenic products.
Right, okay. Yeah, there's a lot of controversy, obviously, at the moment of genetic engineering in all sorts of different areas. But you've asked for my personal view is that we should embrace genetic technologies, particularly in areas such as agriculture and aquaculture. We almost have a duty to do that, but we also need very careful legislation and very careful review about both the safety of individual modifications, health safety and environmental safety. But if we have the right controls in place, then as a society we should be able to understand the dangers and put in place proper safeguards. So that's for animals and plants. If we're talking about genetic modification of humans, which is another area of controversy, then again my personal view is that genetic modification would be very, very useful for somatic changes, so curing diseases, but I don't think we should go near germline modification. I think that should be a complete moratorium against modifying the human germline, but modifying genes in the somatic cells which don't get passed on to the next generation to cure a disease, I think that's a very exciting opportunity in medicine today.
Thank you.
Okay, I think there's a question up there. Is it?
Yes. Hello, Professor Holland. I'm Kenneth, a year-one economics student. So my question is also about genetic engineering. Due to its artificially creating new species as an evolution, and also you've mentioned the agriculture, and I've heard that one of the problems of genetically modified crops is the existence of super weeds and insects. So are there any measures to solve those problems?
Yes. Well, there are two slightly different issues that you've touched on there. One is the evolutionary question and one is the sort of control question. The evolutionary question, it's absolutely right that if we make if the world decides to make a genetically modified organism, whether it be a crop plant or a food item, then we have changed evolution. We have changed its genetic constitution, and that will be inherited to the next generation. That is evolution. We have modified evolution when we do that. controls are important, and it's right to have fears of those issues, as long as we can measure what those risks are. And I really do think that if we can do the correct experiments to measure those risks, then we should, as a society, be able to put in place the correct controls. I mean, the genetic engineering being done today on, for example, some on mosquitoes that transmit disease. There's genetically modified mosquitoes which are transmit sterility so that they can be released into the population and suppress the mosquito population. Very careful controls are put into those genetic modifications to ensure that they don't spread to other organisms and that they can be controlled. So I think we have to go quite slowly in steps with very careful control, but I think experience so far shows that we can do that. Society can do that. It's a responsibility of all society to be involved in the debates and the decisions that are made to regulate those technologies.
Okay, we should have time for a couple of more questions.
Hello, Professor Holland. I'm John from Social Work, Year 1 student. The following question may be a quite silly question. I want to ask about is there really happens in the movie Lucy? Is there really happens about the power of brain? Can we really explore the power of our brain to explore to 10% or maybe 50% or even 100%? Or is it really difficult for human beings to have an evolution of the power of brain?
Right, that's a very, very interesting question. If I had the answer to that, it would be. Yeah. I can only give you a personal view. My personal view is no, we can't. I think we have to think about how evolution works. Evolution doesn't make spare things. Through evolution, natural selection will select for what is necessary for the job that it is doing. So evolution has shaped our brains in a way that is necessary for doing the functions that human beings need to do to survive. Evolution has not generated a lot of empty capacity, in my view. There's just not a mechanism in evolution to do that. It's like evolution evolving a structure which is never needed, and then we have to think, oh, what shall we use that for? So I don't think evolution is capable of doing that, and because from that evolutionary reasoning, I think that the answer has to be no.
Thank you.
Okay, we have the last. Well, there are two more questions. Okay, we can take two. Okay, first up there.
Good afternoon, Professor Holland. I'm To Wei Po from the School of Nursing. Because I do not have much understanding on the molecular biology, so this question may be quite stupid. And I remember the first step of the Darwin's theory is that some variation among individuals in the same species, and then the natural selection happened. And I want to ask, is the variation due to the different arrangement of the genes or even the mutation? If the mutation really matters, that means the DNA between two objects may be very different. So why we can still claim that the two objects are in the same species, but just some mutations there. For example, there is one pig, normal pig, and other one is a pig too, but with wings. But why we can still claim that there is two pigs, but another pigs just have some mutation?
I think the answer to that is really that the scale of the mutations that we actually see in nature are quite small. We don't see pigs with wings. We see pigs with very slightly less hair or something like that. Very small changes. And, of course, we have to remember, we are a good example in this room. Human beings are all different. We are even different from our parents. In one generation, around 50 mutations happen from that we all have. I have 50 mutations in my DNA that my parents did not have. So these varies, and I think I'm the same species. So we have to recognize that most of these mutations are very small, and they have very, very small effects. And it takes a lot of time for natural selection to change one species to another species. So when we're looking in a population of organisms, they will be all the same species. It takes a long time for a population to divide into two species.
So do we have any dominant genes that we can claim that we are still in the same species?
Yeah, even dominant genes will still be will still give traits, so we're in the same species. So yes, mutations can happen with a range of different effects. They don't have to be recessive or dominant, but they will certainly always be considered in the same species. We really only start to talk about different species. The simplest example is when a population of organisms has separated for some reason, such that the mutations that start to accumulate in the two populations over time become different from each other, and if those populations came back together, then they were incapable of reproduction between those populations. So that can happen within one population as well, but it's a slow process and a complex process, but it still involves very small changes.
Okay, thank you very much.
Okay. We'll take the last question. Yeah.
Yeah, good afternoon, Professor Holland. My name is Apollo Lam, Global Studies student. I would like to ask questions not regarding biologies or anything. I would like to ask something rather general. In your experience in teaching and education and research, what do you think is the most important thing that we have to change in the current maybe education system? Because you have given us 3 advices, and one is criticism. And you know that it's somehow hard for us to criticize our teachers or lecturers, professors, because we are kind of trained under a system training us to be obedient or to trust in authority. So what do you think we can do in our education system in order to give us a more advanced or, more modern or a way for us to engage in knowledge in our education system?
I think that's a very interesting question. I mean, when I advise you to criticize, I'm not advising you to criticize everything. Yeah. That's right. I'm trying to advise you to have a framework of thinking which enables you to evaluate the information in front of you and decide which information there's good evidence for and which information there's not such good evidence for, and then think about whether the evidence can be improved or challenged in some way. So to come to how in my experience is a good way to bring that into education, well, in my experience, it's the same in the UK. I mean, for the undergraduate students are not generally feel confident to challenge or criticize a lecturer or professor. So maybe they should, but at the moment they're not. But we can introduce this, at the moment, in my experience, we introduce it at the graduate level, at PhD level. And a good way to do this is to have discussion groups where a group of students will discuss, for instance, a published scientific article, or a published article in a different field will be equally the same. And to go through a scientific paper or any other publication quite carefully and critically, thinking about what the data are and what the conclusions are. And when you do this a few times, you begin to realize that in any field of work, some of the conclusions are quite strong and some of the conclusions are quite weak. Now, doing that, I'm sure that this way of working can be introduced into an educational setting to give people the training in how to critically evaluate evidence and how to think about that. If we can bring this into education at whatever level, I think this will be a very useful and powerful thing to try and do.
Yeah, thank you very much.
Thank you. So Well, thank you for all the questions. Those are all very interesting. Some students said that their questions may be silly or stupid, but I think Professor Holland will agree with me there's no silly or stupid questions, okay? So, all right, so I have to call this to an end, and so we'd like to thank Professor Holland again for this wonderful lecture as well as addressing all the questions.
Once again, thank you, Professor Holland, and thank you, Professor Chu. New Asia College would like to thank Professor Holland for the lecture. So may we invite our college head, Professor Henry Wong, and Professor Wong Kam Po, Director of School of Life Sciences, to present the souvenir to Professor Peter Holland. Thank you both Professor Wong and Professor Holland. May we now invite all professors to take their leave. Students, please remain seated as we will continue our assembly. Now we will have the college announcements. May we invite Professor Chong, Dean of Students. Thank you.
以下落嚟有幾件事要宣佈嘅，咁好多都係上次嗰個雙週會都宣佈過啦，不過再講多次啦。咁大家主要留意下嗰個deadline 嚇。咁第一個宣佈事項就係學生外訪資助計劃咧，咁就都開始接受申請啦。咁呢個計劃主要係資助同學喺暑期到海外參加短期嘅課程同埋學術活動。咁個 deadline 咧就係呢個就係4月。睇下先啦，4月19號星期三下晝5點半。咁第二個宣佈事項就係暑期研究資助計劃亦都係呢個計劃係旨在咧就係倡導學術研究之風同學透過研究項目咧發展個別研究興趣。咁我哋書院學生除咗應屆畢業生之外咧，都可以申請。咁個截止日期都係4月19號。咁第三件宣佈事項咧就係任國榮先生生命科學講座啦。咁第一講就啱啱講完啦。咁仲有第二講同第三講。咁就係第二講咧就係4月8號星期六咁個地點就係喺香港尖沙咀科學館。咁第三講又會講個胚胎嘅就係喺4月10號。咁個地點就係邵逸夫堂。咁同學如果有興趣嘅話就可以參加。第四就係精神健康急救基礎訓練課程資助計劃。咁就呢個係全年接受報名嘅，咁同學只需要完成嗰個課程之後咧，就可以到我哋書院咧就申請翻全數課程嘅費用600蚊。咁第五咧就係學生團體資助撥款咧，就最後限期資助撥款就係5月31號。咁凡請各位新亞書院系會同埋屬會咧喺嗰日之前咧就將所有嗰個資助信同埋所需文件咧就交俾輔導處周玉芬小姐。第六件事項咧就係捐血運動。2017年中大捐血運動咧其實開始咗㗎啦咁到4月13號，咁紅十字會咧就會個流動捐血隊咧就喺4月12號禮拜三即係下個禮拜三咧就上午11點到11點半到下午6點咧就會喺坳群館兩紅基樓林耀明夫人堂協助各位同學捐血。咁同學如果有血多嘅話咧，咁就可以去嗰個捐血啦。咁第七件事項咧就係呢個件事重要啦。17-18年學生宿舍申請咧就由已經開始接受報名啦。咁但係到4月7號 咁就同學local 同埋 non-local 咧都要透過書院咧網頁去申請。咁記住我哋個 deadline 啦。咁暑宿最後又係暑宿啦。2017年暑期申請暑期宿舍申請咧都係4月7號 deadline。咁如果你想住暑宿嘅話咧，咁4月7號之前咧就去申請啦。咁我宣佈事項就咁多啦。
Now we will have the promotion by student organizations. First, may we invite Language and Culture Committee. Thank you.
Good afternoon, everyone. I'm Kenneth from the Language and Culture Committee. I'm here to introduce you a newly refurbished area in the New Asia Canteen, Reading Corner. And I'm now switching to Cantonese. 大家好啊，咁咧我係語言及文化委員會嘅學生代表啦。大家經過 NA Can 嘅時候，有冇見到咧有一個 corner 有咗新嘅顏色啊？咁就係呢一度啦。咁其實咧，如果大家有留意咧，個牆上面咧就有一個 #ReadCrossNA 嘅 logo 啦。咁其實呢一個 area 咧就係俾大家咧去好舒服咁樣睇書啦，或者去做下你平時可能有啲 paperwork 咁樣嘅嘢。咁呢一個咧就係我哋嘅 logo 啦。喺3月31號咧，其實我哋已經正式開幕㗎啦。嗰一日咧，我哋就做咗一啲交換書嘅 party 啦。咁呢一張台上面咧，大家就可以擺低啲書，攞走一啲書。如果你中意嘅，你可以喺度睇都得嘅。咁入面咧，我哋都添置咗好多唔同嘅新書啦。我哋都會定期咧去繼續再去採購嘅。咁希望大家多多參與啦，同埋嗰度咧都有一啲免費嘅書籤可以攞嚟俾大家用嘅。咁同埋咧，牆上面咧就有一個 rules and regulations 啦。咁如果同學去用呢個 corner 嘅時候，都記得愛錫入面嘅書啦，同埋保持乾淨啦。咁最後就係希望大家都 like 翻我哋呢個 page 啦。呢個就係語言及文化委員會嘅 page 嚟嘅。多謝。
May we invite the Student Union of New Asia College? Thank you.
喂，大家好，我哋第54屆新學生會新命啦。咁相信大家入嚟嗰時都收到份問卷㗎啦。咁係一份關於宿生遴選制度嘅問卷嚟嘅。咁大家希望大家花少少時間填一填啦。咁其實而家用緊嘅方案咧就係住所區域45分啦，即係距離分啦。咁住所類型就35分嘅，而最後課外活動就20分啦。咁呢個就係大家今年報宿嗰時會用到嘅方案啦。基本上就大家點填呢份問卷咧，大家今年4月7號呢個 deadline 報嗰時報宿制度咧都唔會變㗎啦。咁但係如果之後下年點變咧，就好睇大家今年而家點填呢份問卷嘅。基本上，咁關於呢個現行嘅方案咧，我哋主要收到學生會收到嘅意見有2個，咁都針對咗呢2個意見咧，俾咗唔同嘅 suggestion 嘅。咁第一個意見咧，我哋就係見到關於住所類型嘅。咁住所類型有2部分啦。第一部分就係關於呢個你住公屋定私屋呢啲咁嘅類型啦。咁第二樣就係關於面積嘅。咁我哋收到嘅意見就係關於呢個你住咩類型，即係公樓定私樓咧。有同學就覺得呢個唔係好真實反映到同學嘅住所需要啦，亦都唔係好多同學攞到呢類型嘅分嘅。所以咧我哋就建議減去咗大部分呢個類別嘅評分啦，而將一啲好特別嘅 case， 即係例如你住鐵皮屋嗰啲咧，就會將你必縮嘅。咁呢個就係我哋針對第一個意見俾嘅 suggestion 啦。咁第二個意見係關於課外活動嘅。咁有同學就認為即係而家住沙田嘅同學咧，無論你係點攞活動，你上幾多支裝，你上20支裝咧，你都係得20分活動分嘅啫。咁就算你爆咗20分咧，都係住唔到宿嘅。咁有同學就反映翻呢個意見出嚟啦。咁所以我哋都俾咗第二個建議，就係將一切照翻今年嘅計算方法去輪選啦，但係預留翻一定數量嘅宿位俾一啲淨係計算活動分嘅同學。即係簡單啲嚟講，即係如果你住屯門，但係你又玩好多活動分嘅話咧，你就會喺第一條隊嗰度就已經篩選咗出嚟作為宿生㗎啦。咁所以如果喺第二條隊獲得宿位嘅宿生咧，就一定會係住得好近中大，而又玩咗好多活動分嘅同學先會獲得呢部分嘅宿位嘅啫。咁關於呢個建議，同學可以喺問卷度填贊成或者唔贊成啦，亦都有個問題俾大家，應該預留翻幾多宿位俾呢啲即係計活動分嘅同學嘅。 咁關於呢份問卷就係咁多。咁另外關於一啲新亞學生會嘅活動咧，咁今晚7點半咧就係呢個新亞迎新營嘅第2次傾莊啦。如果同學想知多啲或者想成為籌委嘅同學咧，亦都記得今晚7點半準時嚟到新亞學生會會室啦。另外何太嘅溫馨提示咧，就係今日嘅5點咧就係 deadline 啦。頭先莊副會長都有講到啦。如果同學係想申請暑期宿舍咧，就記得上網填咗張 form 佢。如果同學係想申請下年嘅宿位嘅話咧，就記得上網填咗張 form之餘喺今日5點咧，就將5點之前咧就將你需要嘅文件交翻去成名本俾何太啦。另外呢個係新亞學生會嘅禮儀晚宴啦，咁就會喺4月10號嘅，就會教一啲學生餐桌關於餐桌禮儀同埋一啲初級嘅品酒技巧，咁就會喺雲喜軒教嘅。咁今日亦都係最後嘅截止日期啦。如果同學有興趣嘅話，就記得快啲報名啦，因為都人數都爆滿咗，不過我哋會採用抽籤嘅形式，所以同學有興趣記得參加。另外咧，4月10號咧就會係呢個 West 暑社嘛。如果同學繼續想喺暑假過3爆4咧，就記得4月10號準時8點半坐低部電腦度打 Q 實殺。另外，如果大家有血多嘅話，都可以跟莊輔導長嘅指示咧，去4月12號喺呢個林耀明夫人堂度捐血嘅。咁另外，新學生會亦都自發喺當日喺練武之地邀請咗神友聯一個器官捐贈組織嚟嘅，咁就會喺練武之地擺一個 booth 去宣傳關於器官捐贈嘅。如果同學有興趣，都可以嚟練武之地去睇下。同埋當日亦都會有器官捐贈卡俾同學寫嘅。如果同學有興趣，都係可以寫嘅。而4月19號就係呢個新亞孫嘅遠征夜話啦，咁就學生會就邀請咗黃乃正教授啦、莊太陽教授同埋陸敏聰小姐去同大家同學見面嘅。同學有咩意見都可以4月19號度發表嘅。另外呢個就係交通組喺新一年嘅開學，即係今年9月會實施嘅第8號線嘅改造啦。咁喺原本8號線呢就會主要係圍繞本部而行嘅，咁新嘅8號線呢就會係由39區開出啦，然後經翻去本部，再兜翻去蘇嗰邊，再兜多次39區，再翻去火車站嘅。如果同學睇呢個圖睇得唔清楚，可以上翻我哋新亞學生會 NASG 個 page 去睇翻我哋個嘢嘅。再最後咧，因為 show 唔切張 slide 啦，咁我哋其實新亞嘅人字拖咧已經到咗啦。曾經喺呢個 CU Secret 掀起風雲嘅新亞人字拖咧，大家而家已經喺呢個可以上去我哋新亞學生會嘅會室度購買噶啦。宣佈上係咁多，唔該晒。
This is the end of the 597th Biweekly Assembly of New Asia College of CUHK. Students, please be reminded that before you leave, you must return the completed attendance sheets with your name, major of study, year of study, and student ID number clearly written on to the collection box for record of attendance and collection of opinions. Thank you for your cooperation.